I would like to thank the organizers for the opportunity to present today and also for putting together this amazing conference. So I'm Francesco, if you want to say it well, and I come from the old continent, and I would like to present you my work, which is about neuroimmune communication in response to light. So light is a very primordial physical entity of our universe, and has accompanied life basically from the beginning. In fact, it was there before life, and it will very likely be there after. And this, of course, comes with not surprise that all kind of organisms basically have evolved specialized structures that are able to respond to light. And these are called photoreceptors. And these can be proteins in simple unicellular organisms, or specialized cells in more complex organisms. In mammals, the organ that is the photoreceptive organisms is the retina, and we can see from the anatomy of the retina here that light, that light comes from this side, goes across all the cells and hits the photoreceptors here, which are called rods and cones. This is a nice image from Santiago Ramón y Cajal from the beginning of last century. I always like to show this. And so what happens is that we always thought that these cells were the only ones that were photoreceptive. However, recently, some of these neurons here, which are the ones that make the optic nerve, have been shown to be intrinsically photosensitive. That's why IPRGCs, which means that there is not only these photoreceptors, there's also these ones. And so if I were to ask you what is the role of the eye, what would you say? Vision. So we use the eye to see things. That's true, but you know, there are a lot of different, this is true because there are a lot of neurons, a lot of RGCs that project to visual areas, but there's also a lot of non-image forming neurons that induce a number of non-image forming actions. And we can see that the majority of these non-image forming actions are performed by IPRGCs. So recently, a number of studies highlighted the importance of these non-image forming pathways, and this, for example, is true in mood, is true in learning and memory, glucose metabolism, energy metabolism, and so on. And this, prompts us to ask whether light was able also to affect immunity. So does light directly affect immune responses? And does it do it via a neuronal circuit that connects the brain to immune organs? And finally, what are the cells that are responding to light? And so to reply to this question, to investigate this question, we propose the following model, which is depicted in the diagram here. We have lymph nodes, which we learned yesterday are very important key immune organs. They are richly innervated, in fact, and they have sympathetic and sensory fibers, are shown recently and reviewed by us also. And we propose that lymph nodes are directly connected to the retina via neuronal connection that goes through the brain in the locus ceruleus that, again, we learned yesterday to be a very important gateway between the brain and the body. And then from there, through an efferent sympathetic pathway, activate lymph node or modulate lymph node activity. So first thing, we took some mice from in the dark. They were in the dark. We exposed them to light for an hour and screened for a number of immediate early genes in lymph node. And we can see I'm showing here MYC as it's because we don't have time to show all of them. And we can see that MYC expression is increased in all lymph nodes, but not in spleen. These are lymph nodes that come from different regions of the body. And we also see that in a time course analysis, 1 hour is where MYC expression peaks. Importantly, when we performed this same experiment during the light phase, we are able to see the same activation of MYC in lymph node. And also, when we used red light, for which mice do not have the receptor, this activation is abolished. And this shows that light is able to activate specifically the lymph node. But of course, as I said, light is perceived by the retina. So, and the effect that we are studying is clearly a non-image forming effect, as far as I can tell. So we hypothesized that probably iPRGCs are important for this effect, so that we deleted iPRGCs genetically, and we see that when we knocked out these cells, we lose the effect of MYC activation, showing that these cells are important for lymph node activation. So now I showed you that lymph nodes are activated by light, and the retina is important for this effect. So what is in between? Well, first thing that we did is to see whether this effect was systemic or was due to local innervation. And for that, we blocked nerve transmission specifically with lidocaine, only on one side, using the contralateral side as a control. And what we can see, remarkably, is that this was important. So lidocaine treatment abolished light-dependent MYC expression when compared to the contralateral side. So next we asked whether that was due specifically to sympathetic fibers. And for that we injected 6-hydroxydopamine, that is a toxin for sympathetic fibers, so that we deplete them. And after light stimulation we see the same pattern. We see that 6-hydroxydopamine was able to reduce MYC expression in the lymph node. We also went one step further and asked whether the signaling downstream sympathetic activation was important, and for that we used alpha adrenergic receptor inhibitor and beta adrenergic receptor inhibitors in combination with light stimulation. And we can see that in both cases we have a reduction of MYC expression in here. So together, these data are able to show, we are able to show that sympathetic signaling is important for lymph node activation. So next, we wanted to study what are the intermediate relay stations. So we go one step further. For the ones of you that are not familiar with sympathetic system, the sympathetic system is made of two neurons. One neuron that is the postganglionic neuron, that is the one that innervates the target, and one that is called preganglionic neuron, which is before the ganglion, and as the cell body in the spinal cord, specifically in a nucleus that is called intramediolateral, IML for short. And here we see a coronal section of the spinal cord, and in cyan we have chat that marks the IML, and FOS in magenta is a proxy of neuronal activation. And we can see that in this close-up, that after light stimulation, we have an increase in FOS expression in chat-positive IML neurons. And the same was true for the locus coeruleus. We stained locus coeruleus neurons with TH. TH marks catecholaminergic neurons, in this case, locus coeruleus. And again, CFOS as a proxy of activation. And we can see in the close-up, highlighted by the arrows, that FOS expression is increased in TH-positive LC neurons. So to further prove that locus coeruleus was important for this effect, we performed optogenetic stimulation of the locus coeruleus. So optogenetic stimulation is basically, to do optogenetic stimulation, you infect neurons with a virus that carries a channel that is activated by light. So after putting a probe that has an LED at the end, we are able to specifically activate or inhibit, depending on the virus that you choose, the neurons that are infected. And in this case, we were activating the locus coeruleus for 1 hour, and we can see that after 1 hour activation, MYC expression goes up in lymph node, but not in spleen, corroborating the light stimulation data. So now, The locus coeruleus is a tricky guy. As yesterday, we also know that it was important for lymphatic system, is important for stress, for arousal, and a number of different factors. And does this via is many different connections. It's connected basically to all the areas in the brain that you can think of. But we are, interested specifically on one single connection, which is the descending pathway from the locus coeruleus to the spinal cord. And to specifically target this pathway, what we did is to inject a virus in the spinal cord that carries a cre-dependent DREAD inhibitory receptor. This was done in DBH mice. DBH is expressed in catecholaminergic neurons, noradrenergic specifically, and in the central nervous system, this means locus coeruleus only. So therefore, this approach allows us to mark specifically or to modulate specifically the descending ceruleospinal fibers that from the locus coeruleus innervate the site of injection in the spinal cord. So then we injected CNO in order to activate the virus, and after we did light stimulation, and this is an example of the fibers that you see in the spinal cord. And after stimulation, we see again that in the control, MYC goes up, but when we inhibit specifically the fibers, MYC expression is abolished. And this clearly shows that these fibers are important for the effect. So finally, we wanted to investigate what are the cells that are responding to light, and for this we performed single-cell RNA sequencing from the lymph node upon light stimulation. Here I'm showing you a UMAP. We can see that we are actually able to get a lot of different cells, which is not obvious from lymph node. These are myeloid cells. These are lymphoid cells, which is, of course, the vast majority, as well as stromal cells, which are notoriously difficult to isolate. And unfortunately, we are currently analyzing this data set, and I will not be able to tell you what are the cells that respond to light, maybe in the next HPFS conference. And this brings me to the conclusion, unfortunately. So I showed you that the light is able to modulate lymph node activity via an efferent signaling, sympathetic signaling, that the retina, locus coeruleus, IML are involved in this pathway. And open questions remains. First of all, what is the first responders? What are the first responders? What are the cells that in the lymph node respond to light? And also, can we use light to improve treatments? And this may be for a future HFSP grant. I'll just put it there. With this, I'm done, and would like to thank the lab, our collaborators in Europe and overseas, and thank you for your attention, and happy to get any question. That was a good timing? Yeah. Yes.
Great talk. I just had a general question. So is this mechanism just complementing other like the rest of the immune system, or is it 50%, 30%? Like, I'm just trying to understand how it's impacting the lymphatic system. Is it supplementing the natural process, or?
Great question. The simple answer is that I have no idea. So if you want me to speculate a bit, what I think is happening is that mice are nocturnal animals, no? And we know that the immune system is particularly active during their rest phase, so during the day. So what's happening is probably an evolutionary conserved mechanism that allows the immune system to be primed to respond to light, because light is something that we know that is gonna come up at some point. and to prime up, as I was saying, these cells to be active during the rest phase of the mice. This is one possibility. The other possibility is that if it's not immune cells if we will not find that immune cells are modulated by stromal cells, for example, endothelial cells or like vasculature in general, which is also possible due to the anatomy of the innervation. So if you look into a lymph node and you stay in the lymph node with a marker for sympathetic fibers, you will see that they run along the vessels. So is it very possible that that vessels will be the first responders, which also be, it can go in line with what I said before, or can be a different mechanism. So the short answer is that I don't know. I don't know yet.
Have you done any experiments with blind mice?
No, I'm doing it now. Yes,
okay.
I'm doing it now, and I think so. I just showed the one in knockout mice, which is kind of similar. In that case, we are able. In this case, we are able to specifically deplete cells that are photoreceptive. In the blind mice, the experiment that I will do with a crash of the optic nerve, we will of course have no signal from the.
Exactly. Yeah. Okay. Thanks.
It's coming. It's coming. Thanks.
Fabulous talk. So the sympathetic nervous system also innervates the bone marrow, right?
Sorry, can
So
you?
the sympathetic nervous system also innervates the bone marrow, right?
Yes.
Do you see also changes in the hematopoietic stem cells and their behavior? And the other question was, because the hematopoietic stem cells move in and out of the bone marrow and follow a day and night rhythm, right? So I was just curious. And the other thing is that, do you see that they have differences in their immune responses? Are they better or they worse? I mean, when you abrogate this cascade that you're talking about.
So to reply to the first question, the bone marrow, the answer is yes, meaning that in the first screening that I did see some genes that were modulated also in the bone marrow. I do not have the blood data yet. I actually collected blood from these mice, but I still didn't analyze it. So this is something that will be probably done soon in the future. It's definitely, I mean, the sympathetic nervous system innervates virtually all organs.
Exactly.
Also, like spleen, for example, is another organ that I took and in which I see different. I don't see MYC expression going up, but I see, for example, FOS expression going up after stimulation. And the last question was?
With respect to immunity, does it actually influence their immune capabilities? Are they better or worse? Or
So the answer is, in the literature, is yes. Specifically in immune system, the most expressed adrenergic receptor is beta 2. This is like well known, well established, and what it does when you activate it, what happens is that you modulate the migratory capacity of some of these cells. Now, I do see differences also with alpha. Alpha usually expressed on the vessels, so that. Again, I don't know yet. I will. I will see soon. I will see soon which are the cells that are the first responders at least, and then what happens in the with longer exposures or longer time points.
Great, thank you so much.
Marco.
Thanks. Yeah,
I talk too much. I'm sorry.
No, it's a really stimulating talk. I had a question, which is that I mean, the idea that you're proposing is, I think, quite remarkable, that there's like direct neural connection between the visual system and the immune system. Is like a potentially like a more economic hypothesis is that a lot of cells have their own circadian machinery. They have like the Frick proteins, and that can create its own sort of independent timekeeper that becomes set by the diurnal cycle, but then can act independently. So would a critical control of your hypothesis be to try to knock out the circadian proteins and see if things still work the way that you are showing, or would that just be too disruptive of a change?
So this is a great question, and I'm sorry I'm going to be a bit long maybe to reply to this, because I think it's a complex system. So the first answer that I can give you, like very short answer, is that I don't think that this is a circadian effect at all, for two reasons. First of all, because it's too short, and second, because I actually showed here You can see this is the stimulation during dark phase, and this is the stimulation during the light phase. So just 12 hours apart, showing that there is no time of day difference. You have the same thing, so there is no time of day difference. And then for the circadian system, so circadian system this is something that it's usually confused with light in a way. So the circadian system is self-sustained, does not need light. It can be entrained by light. So actually the two things are separated. This does not mean that circadian genes may influence light responses. Actually, it's known, for example, that PER2, period 2, is one of the most critical circadian genes, is a strong responder to light. In the SCN, which is the central pacemaker, if you shine light on the mice, you have PER2 expression going up. But why? Because they know that when you give light, we are ingrained in a way that if you give light to them, they know that they have to go to sleep basically, and so something happens. But I don't think it's a circadian effect, and I don't think that knocking out circadian genes will necessarily be a problem. I'll probably do it at some point. I will, reviewer 3 will ask, can you knock out the new gene? And we'll do it, but I think for the moment, these experiments prove that it's not time of day dependent. Thank you. Yes?
Have you tried any different wavelengths of light?
Not yet. So the only thing that we tried is red light, because mice do not have red light. And if you want me to give a bit more details on this experiment specifically, so we did this because as mice do not have the receptor for red light, we wanted to test whether it was the manipulation that we were doing to affect, like a stressor kind of thing. And I don't think so. So because of course you don't have it. The nice thing about the iPRGCs is that they are selectively responding to blue light. So we can be able to. I just got this data like last week, literally. So so it's very new. I will definitely do the experiment showing shining blue light on them and see if this, because this is of course important for therapies. There are a number of bright light therapies in which they use blue light, and they see some improvements in, for example, for depression and so on. So I think this could be a potential mechanism. Thank you. Thank you.